Thank you all very much for joining me for this webinar. I'd like to start by just giving an overview of what I'll be discussing in this webinar. I'll start with an introduction to chromatin structure and modifications, followed by a brief overview of the basic ChIP protocol. Next, I'll briefly introduce the histone code hypothesis that was first presented in 2000. Then I'll go over what data from ChIP qPCR looks like, and then review some interesting publications that use chip-on-chip to analyze chromatin modifications and genome organization. I'll then discuss some publications where ChIP-seq has been used, and finish by explaining some recently developed methods that use variations of the ChIP protocol. So, what is chromatin? Well, in the human genome, there are around 3.2 billion DNA base pairs, which if unraveled, would stretch to around 5 feet of DNA in every cell. To remedy this, DNA is tightly compacted into a higher order structure called chromatin, so that the DNA can fit inside a cell's nucleus. Chromatin, as we'll soon learn, is also an important factor in controlling gene expression. The most basic unit of chromatin is the nucleosome, which consists of DNA wound around a core of 8 histone proteins. The nucleosome core consists of approximately 147 base pairs of DNA wrapped around the histone octamer, consisting of 2 copies each of histone H2A, H2B, H3, and H4. Histone H1 sits on top of this structure to keep the wrapped DNA in place. This beads-on-a-string structure is called euchromatin. When multiple nucleosomes condense even further to wrap into the 30-nanometer fiber, this structure is termed heterochromatin. Finally, there is even higher level packaging to form metaphase chromosomes during mitosis and meiosis. The histones I mentioned earlier can undergo many post-translational modifications that affect how they interact with DNA, some of which are shown here. Histones H3 and H4 are the most commonly modified histones and have long N-terminal tails protruding from the nucleosome whose residues are targeted for acetylation, methylation, phosphorylation, deamination, and ubiquitination. There are also modifications to histone variants, such as phosphorylation of histone H2AX at serine 139 that serves as a marker for DNA damage. Specific modifications have different effects on gene transcription, as we'll see later. In order to determine which histone modifications, as well as where other DNA-protein interactions, such as transcription factor binding, are present at certain regions of the genome, chromatin immunoprecipitation, or ChIP, was developed. In ChIP, DNA is cross-linked to proteins using formaldehyde, and the DNA is then fragmented by sonication into fragments of 200 to 1,000 base pairs. In some instances, when the binding of protein to DNA is stronger, such as with nucleosomes, cross-linking is not necessary, which is referred to as native ChIP. In native ChIP, micrococcal nuclease is used to digest the DNA, which produces DNA fragments the size of a monosome. Next, a specific antibody of interest is added to bind to the protein-DNA complex, which is then immunoprecipitated, pulling down only DNA fragments that are directly bound to the target protein. After several rounds of washes, crosslinks are reversed if applicable, protein and RNA are degraded, and the DNA can be purified for later analysis. The abundance of DNA pulled down can be determined compared to the input, or the starting chromatin amount. So now that we're familiar with ChIP, I'd like to present an early ChIP experiment from 1988 that was one of the first studies to identify a link between histone acetylation and transcriptional activation. Here, the authors developed and categorized an antibody raised in rabbit that was specific for acetylated histone H4. chromatin was isolated from erythrocytes of 15-day-old chicken embryos and was treated with micrococcal nuclease to fragment the DNA. After incubation with the antibody, the immunoprecipitation was carried out using formalin-fixed Staphylococcus aureus cells that had an active protein A in the outer membrane, so this was before there were protein A or protein G beads. They then dot blotted the DNA and performed a southern blot for alpha-D-globin with chromatin from 3 stages of the experiment. Alpha-D-globin is actively transcribed in these cells, and the authors found a very high alpha-D-globin signal in the bound fraction, which contained the antibody for acetylated histone H4, as you can see in column 5, demonstrating the presence of acetylated histone H4 at a very active locus. You can also see that the input, unbound fraction, and fractions that contained no antibody did not produce alpha-D-globin signal.